Welcome friends. After a long interval, welcome to the biology classes. Today we are going to discuss a very simple chapter, that is nervous system. We had our knowledge that nervous system is made up of nerves. Nervous system is made up of nerves. So, and we have come across that nerves are made up of neurons. And we know that neurons are having a cell body. This is cell body. These are all dendrites, and this is known as action. This much we have come across. Now let us give a little introduction why this nervous system has developed. Initially, animal had a single cell. So this single cell having one nucleus, they were capable enough to do all the activities within this cell. So there was no need of this nervous system. But as and when, because of the evolutionary process, the single cell animal has become a multicellular animal. A multicellular animal. So we are multicellular. These multicellular animals have numerous cells, and there are different type of cells doing different type of activities. So it was imperative that to develop a system to control and coordinate control and coordinate between various cells. So in order to control and coordinate the different types of cells in a multicellular animal, there was arose that there should be a system. So evolutionary process, nervous system has developed. Another system developed known as endocrine system. Endocrine system. Both are for control and coordination. But these are very slow, and that we will discuss in our endocrine chapter. But this nervous system is very quick in response, so the nervous system has developed accordingly. This is a short introduction. And nervous system is a very simple system because the nervous system is made up of single type of cells known as neurons. So there is no other cell. You don't have to go through different type of cells. So nervous system is made up of neurons. Besides these neurons, there are some supporting cells. Those supporting cells are known as astrocytes, oligodendroglia, and microglia. These are the three supporting structures. These are all very small cells. Microglia is the smallest one. Very small cells having these processes and others. So these are the supporting cells of nervous systems. But the main nervous system consists of neurons. So neurons, we know that neurons is a cell body having various processes. This is a cell body and these are the processes. The receiving processes are known as dendrites and the acting or motor acting process is known as action. And this is grey in color. Whereas these dendrites and actions, they are white in color. Sometimes we find that there are some medullary sheath over these fibers. So these are called medullated nerves. And those who do not have this medullary sheath are called non-medullated nerves. Our nervous system mainly consists of central nervous system, CNS, and peripheral nervous system, PNS, peripheral nervous system. In the peripheral nervous system, there is autonomous nervous systems. That is not our discussion today. We will discuss on the central nervous system, then peripheral nervous system, then we will go to autonomous nervous system. Now, the central nervous system consists of, this is the brain and this is the spinal cord. This much we have come across. The central nervous system consists of brain and spinal cord. Very simple. This brain is enclosed by the skull or cranium. It is covered by skull or cranium, while the spinal cord is covered by these are called vertebra. So within the vertebra, this spinal cord goes inside. So this brain and spinal cord are the constituent of central nervous system. Now, other than the central nervous systems, the nerves which goes away have some covering. This covering are known as neurolemma. So other than central nervous system, whatever the nerves are there, nerves are covered by a covering, nucleated covering called the neurolemma. Okay. Now we'll go into the detail of this brain and spinal cord. But all these are consist of neurons. Now, this brain, we should consider it simple structure. Brain and spinal cord. Suppose this is let me draw it more clearly. Brain and spinal cord, I have cut the brain into two halves. like gourd, Gourd. Gourd you have seen. You cut the gourd in two sides. So this is the half portion, this is the another half portion. Okay. Now this brain is very precious because it is made up of neurons. Neurons who are lost cannot be reclaimed, cannot be regenerated. Now this brain should be protected. So it is protected by cranium. Other than this, it should have more and more protection. Let's see what are those protections. The brain has three coverings. Brain and spinal cord. Both are having coverings. This covering is known as pyometer. Other than pyometer, there is another covering. This pyometer which remain attached with the surface of the brain. And spinal cord. There is no space. So this is pyometer, and above the pyometer there is a little bit space, and this space this covering is known as arachnoid matter. Clear? First pyometer, this is arachnoid matter, and above this there is another covering this covering is known as tough. This is fibrous dura matter. So this is dura matter. There is a space. This is arachnoid matter. There is a space, and this is pyometer. These do not have any space. This covers. So pyometer is like banyan or Genji, Lux cozy. So this is a Genji or banyan like structure, it remains covered. So above this there is sharp, this is sharp. So this sharp and banyan, they have some space. This space, since it is below the arachnoid, this is known as subarachnoid space. Clear? And this covering is good. Good. So there is a space. This space, since it is below the dura mater, it is known as subdural space. And this is known as subarachnoid space. Subarachnoid space. So in between these two remains filled with CSF. CSF means cerebrospinal fluid. It is a fluid like plasma. This CSF remains filled here. So this is water. Say for example, this is watery substance. And this also remains filled with CSF, this space. This also remains filled with space, subdural space. So in the subdural space, there is fluid and here is also fluid. So it protects above this, there is cranium or skull. Skull is made up of flat bones. So this skull, if there is heavy jerking, this brain, that is neurons, made up of mainly neurons and oligodendroglia, that three supporting cells. These neurons or brain matter do not get collided with this cranium. So it is protected by two layers, that is subarachnoid space filled up with water and subdural space filled up with water or CSF. So it is protection. Above this, above the skull there remains the subcutaneous tissue. And above this remains scalp, where from the hair grows. So above the scalp there is hairs. So it remains first hair, then scalp, then subcutaneous tissue, then skull, cranium, then dura mater is tough, is fibrous. It is fibrous, it is tough. Then there is fluid, then there is this matter, pia matter, and this is nerves. Not only this, it contains CSF inside also. How? Let us see what are those are called ventricles. Suppose here, this is like a gourd, which I told you earlier. This gourd, I have cut it here. I have taken some neurons and I have made a ditch here. And I have also made a ditch here. So there are two ditches. These are called ventricles, these are hollow. These ditches joins to a third ditch. This is also ditch. I have taken out some flesh or taken out some neurons from here by teaspoon. Teaspoon I have taken neurons. I have taken neurons from here, neurons here. So it remains semicircular ditches. These are called ventricles. This is say first ventricle, this is second ventricles, or these two are called lateral ventricles. And this ventricle is known as third ventricle. Again, I have taken a semicircular canal here. That is, I have taken out some neurons from here itself. It is semicircular like this, semicircular. So I have done ditches here so that the CSF, it remains filled with CSF. It comes here. Then again I have taken some flesh or some neurons from here. I have made a ditch here. And it continues here by semicircular canal like this. Like semicircular canal, I have made a ditch here, semicircular, up to this. Now, if I made the same ditches here, and I place these two in a single gourd, like a single gourd, so it will be hollow, because this will fit here, this will fit here, this will fit here, this will fit here. So inside it is hollow and remains filled with CSF. So these ventricles also remains filled with cerebrospinal fluid. Now this cerebrospinal fluid is inside and cerebrospinal fluid in two layers outside. So if you jerk it, if you jerk like this, even if you jerk very hard, it does not hamper the neural cells or nerve cells. These are called lateral ventricles. This is third ventricle, this is fourth ventricles. So ventricles remain filled with CSF, as well as these two, subdural space and subarachnoid space also remains filled with CSF. Now, there are some terms which we should remember, because sometimes it comes in your exam. These two lateral ventricles remains joined by canal. This canal, this canal. These two canals known as foramen of Monro. These two lateral ventricles remain joined with the third ventricle by foramen of Monro, named after the scientist Alexander Monro, Scottish physician. He has discovered it. So foramen of Monro, or these are called joining canal. Now this is third ventricle. Third ventricles remain joined with, this is half, and that half it remains hollow. So this is known as aqueduct of Sylvius. Clear? Or this is known as cerebral aqueduct. Cerebral aqueduct. Clear? Now this goes to fourth ventricle. The CSF passes on from here, and it continues with here. This is the neural canal, central canal or neural canal. The question may be asked that where from the CSF comes? Is it not? So let us consider that this brain is filled up with the coronary artery or carotid artery, internal carotid, external carotid, those arteries. Actually, here there are blood sinuses. This pyometer, durometer like this. Here some blood sinuses are there. From artery, from heart, it comes here like this, and it innervates here, in this way. It comes, and the plasma are formed. Plasma, these are known as CSF. Clear? In this way, blood comes. Now, again, question comes that the ventricles will filled with what? CSF. Where from? This space is known as subarachnoid space. Where from the subarachnoid spaces gets CSF? There is no vacant places here. Where from the subdural spaces gets CSF? Clear? Now let us go for that. This I am taking out, this fourth ventricle. fourth ventricle is like this, and it continues within the spinal cord. So this is fourth ventricle. It joins with the cerebral aqueduct or aqueduct of Sylvius. It gets here. In the fourth ventricle, in the roof, this is roof of the fourth ventricle, there is one aperture. That aperture is known as foramen mesendae. Foramen mesendae. This foramen mesendae is here, and there are another two openings on both sides of the fourth ventricle. Here one, here one. These are known as foramina or Lusca. Foramina or Lusca. So this, from this foramina mesendae, which is on the roof of the fourth ventricle, and from the foramina of Lusca, which is present on both side walls of the fourth ventricle, from this area, the CSF, from this area the CSF passes on to this space, this space, this space. So what is this name of this space? This space is known as subarachnoid space. Now it is over. That subarachnoid space gave the supply of CSF from foramina mesendae and foramina of Lusca. Clear? Now where from here it goes, there is no spaces. Subsequently, we should say that subdural space get the supply of CSF from this arachnoid. This arachnoid space has some granulations. Some granular spaces are there. So these are called subarachnoid granulations. The subdural space receives the supply of CSF from arachnoid granulations. Now it is there will be no question that these foramina of Lusca and foramina of magnesite supply the CSF to subarachnoid space, and subdural space got the supply from the arachnoid granulations. Clear? Now from the inside and outside, brain inside is hollow, full of CSF, and outside is CSF. So it does not go here and there. If you put a balloon in a floating area, balloon will go here and there. But if you put some water inside, it will be there inside. Whatever, as per your choice, it will be there. So in this way, it is filled up with this. Now, whenever foramina or foramen of Monroe, sometimes question comes, what is foramen magnum? Foramen magnum. It comes in the exam. What is foramen magnum? Foramen magnum actually, let me draw a little bit. Suppose this is our brain. This is the frontal lobe, this is the occipital lobe. Lobe will come in the skeletal system. And the last lobe, this lobe is known as occipital lobe. occipital bone. That lobe and this bone is known as occipital bone. This occipital bone has a oval opening through which the spinal cord emerges, passes through the vertebral column. In the vertebral column, the spinal cord passes through. Vertebral column is hollow. This vertebra has hollow structure. The spinal cord comes here. These are the spinal cord. So the spinal cord passes out from the occipital. There is a hollow area, oval shaped hollow area at the base of the occipital bone. This bone is known as foramen of magnum. Clear? So question comes, what is foramen of magnum? Then you will say it is a oval shaped area, space in the occipital bone of the skull through which the spinal cord emerges, called as foramen of magnum. Foramen of Monro, this is the joining of the two lateral ventricles with the third ventricle. Then another question comes, have you heard it not? Foramen ovale, O-V-A-L-E, ovale. Link. What is foramen ovale? Nothing. Before birth, this is left. So this left auricle, left ventricle, right auricle, right ventricle. Now, left auricle got the oxygenated blood from the lungs. From the lungs it comes the oxygenated blood here. And before childbirth, so the oxygen supply of the baby is done by the mother, taken care of by the mother. Because the lungs, which is going to lungs, this is the impure blood, as in adult we have seen, this is impure blood, it goes to lungs. From the pulmonary artery it goes to lungs. From lungs, oxygen, carbon dioxide goes out and oxygen comes in. But in child, before birth, there is no need of this, because they get supply, the baby gets supply of oxygen from the mother. Mother take care. Moreover, this remains filled with amniotic fluid. So what actually happens, whenever blood comes here, it bypasses from here. Instead of going to lungs, it bypasses from here to different parts of the baby's body. So this space, there is a hole. This hole is known as foramen ovale. Clear? So foramen Monro, foramen magnum, foramen ovale, foramen magenti, and foramen of Luska. All the foramens, you write it down. Now what happens after childbirth? It remains, it gets like this, foramen ovale, after the birth of the child. Within 6 months to 1 year, 6 to 12 months, it gets filled up. If it does not, it remains a condition known as patent foramen ovale, PFO. In short, it is known as PFO, patent foramen ovale. So doctor do not usually go for surgery. They give some anti-clotting agent. anti-clotting agent so that the blood does not clot. Number one. Even if it persists, along with other defective conditions of the heart, they go for surgery. They catheterization these or stitch these in the heart. So this is known as foramen ovale. So we have come across so many things. Let us write it down again. Number one, foramen. Monroe. Number two, foramen. Monroe foramen magnum, foramen mesendae, foramina oblusca, and another one, foramen ovale. This you go through, you search where, everywhere you will get right short notes in this way. So this is known as, this is one part of the brain. Till today we will conclude here, and then the next day we will go for nerves, various nerves, cranial nerves. In order to remember it, there is a technique I will tell you. Peripheral nerves and cranial nerves and spinal nerves we will discuss in the other time. Till then, goodbye, take rest. Thank you very much.